Welcome to the C2 Pipeline Innovation and Curiosity Center Presumptive Blood Testing and Blood Typing Lab. By the end of this session, you should be able to do the following. Understand what makes up your blood, understand the different ways that blood can be detected, understand what agglutination is, be able to determine the blood types of the samples provided to you, and determine the next step if a match is found among the samples. So just what is it that makes up our blood? Plasma. This is the yellowish liquid portion of blood that contains electrolytes, nutrients, vitamins, hormones, clotting factors, and proteins such as antibodies to fight infection. Red blood cells, also called erythrocytes, are the most abundant cells in our body. They are produced in the bone marrow and contain a protein called hemoglobin that carries oxygen to our cells. White blood cells, also called leukocytes, are part of the immune system and destroy infectious agents called pathogens. Platelets, also called thrombocytes, are the clotting factors that are carried in the plasma. They clot together in a process called coagulation to seal a wound and prevent a loss of blood. Here are some blood facts. The average adult has about 5 liters of blood in their body, which makes up about 7 to 8% of their body weight. Blood is a living tissue that carries oxygen and nutrients to all parts of the body and carries carbon dioxide and other waste products back to the lungs, kidneys, and liver for disposal. It also fights against infection and helps heal wounds. There are about 1 billion red blood cells in 2 to 3 drops of blood. For every 600 red blood cells, there are about 40 platelets and 1 white cell. Let's find out how blood is detected. There are 2 major chemical tests for the presence of blood. They use luminol and fluorescein. As seen here in the picture, there is a fingerprint that's visible using luminol. Also, another picture of a shoe print that is made visible using the chemical luminol. Luminol reacts in the presence of heme, which is part of hemoglobin, and undergoes chemiluminescence as seen in the pictures. Fluorescein, another chemical test, undergoes fluorescence and a strong short light wave is used to induce fluorescein. It also includes a thickening agent that allows it to cling to vertical surfaces, such as this handprint that was on a wall. As you can see in the picture of the inside of the van, the light wave and fluorescence has detected large amounts of blood in this vehicle. There are field tests for testing blood outside of the lab. The Kastle-Meyer test is a presumptive or screening test to determine if a substance is blood or is not blood. Phenolphthalein, which is a color indicator, is mixed with hydrogen peroxide, which combines with the heme in the blood. If the test is positive, it gives a bright pink fuchsia color and no color at all if the test is negative, as seen in the picture. Another field test for detecting blood is the hexagon OBTI test. This is a test to determine if blood is human or animal. It is similar to a home pregnancy strip. The absorbent strip contains blue particles and monoclonal anti-human hemoglobin antibodies, which determine your immune system response to allergies and infections. The control indicator, which is labeled C, has a blue stripe to indicate a valid test. The test indicator, which is labeled T, has a blue stripe if the sample is human. If there is no blue stripe, the sample is from an animal. Now we will learn about blood typing. Let's take a historical look at blood typing. Around 1900, Karl Landsteiner discovered that there are 4 different types of blood based on the presence or absence of specific antigens found on the surface of red blood cells. In 1940, Alexander Wiener and Landsteiner reported the discovery of the Rh factor by studying the blood of the rhesus monkey. 85% of Caucasians, 94% of African Americans, and 99% of all Asians are Rh positive. Genetic markers in blood are as follows. Not all human blood is the same. The most familiar of the blood group system is ABO. There are 4 subgroups in the ABO system. They are A, B, AB, and O. Each subgroup is characterized by the presence of certain antigens on the surface of red blood cells. Agglutination occurs when antibodies and antigens come together. Agglutination. This will be an important part of our lab. Agglutination of red blood cells occurs when 2 non-compatible types are mixed. Compare the 2 pictures at this time. As you can see in this picture, the blood cells are not gathered together. However, in this picture, the cells are clumped together. When the cells clump or clot together, this is called agglutination. You will be looking for this in our lab today. Here is a model of the ABO group. Blood type A, as seen in this picture, has antigen A on the surface of the cell and will agglutinate or clot with blood type B because it has anti-B antibodies. Take a moment to study the other blood types. This is blood type AB. The blood type AB has no antibodies and does not agglutinate with type A or type B blood. As shown here, blood type O has no antigens on the surface of the cell and will not agglutinate or clot. Let's take a moment to study this chart. This chart shows which blood types can give blood to other blood types and which blood types they can receive blood from. As you can see, blood type A can give blood to blood types A and AB. Blood type A can receive blood from people with blood type O and blood type A. During our lab today, we will be studying Rh factors also. Scientists sometimes study rhesus monkeys to learn more about the human anatomy because there are certain similarities between the 2 species. While studying rhesus monkeys, a certain blood protein was discovered. This protein is also present in the blood of some people. The presence of the protein, or lack of it, determine whether the blood type is positive for Rh or negative for Rh. This is referred to as Rh for rhesus factor. We will now begin our experiment.